Mensen aan en uitzetten.
Ja, ik denk het.
Ik kan niet chatten.
Dus even kijken. Chatten, je bedoelt? Wij zullen wel iedereen installeren aan het begin dat hij zichzelf moet muten,
kan wel mensen aan en uitzetten.
de mensen die jij aanspreekt. We zijn met jury, we zijn met Jasmine en Hassan. En die kan jij het woord geven. Ja, ik denk het.
Maar iedereen anders kan ook vragen stellen, neem ik aan.
Chatten, je bedoelt?
Als het zover is.
Wij zullen we beginnen dus zelf met muten vanuit jou. En de mensen die jij aanspreekt. Vanuit het publiek zijn, dan kunnen deze gesteld worden. We zijn met Jasmine en Hassan. En die kan jij de woord
Ja, ik denk het. Doen jullie de timing? Maar iedereen anders kan ook vragen stellen, neem ik aan. Als het zover is.
Wij zullen beginnen dus zelf met muten vanuit jou. En de mensen die jij aanspreekt. Vanuit het publiek zijn, dan kunnen deze gesteld worden. We zijn met Jasmine en Hassan. En die kan jij de woord
Ik ben kennelijk geen teammember, dus. Doen jullie de timing? Maar iedereen anders kan ook
alleen de mensen die
vragen stellen,
zelf
neem ik aan. Als het zover is.
wij zullen beginnen dus zelf met muten
Dan kan je niet
en de mensen die jij aanspreekt. Kunnen deze gesteld worden. We zijn met Jasmine en Hassan. Maar als je gewoon het die
ik ben kennelijk geen teammember, dus. Doen jullie de timing? Maar iedereen anders kan ook
alleen de mensen die zelf
zover is.
maar wij zullen dus
Dan kan je niet
tijd in de gaten houden. En dus straks ook zeggen wanneer de volgende is beginnen deze discussie. Maar als je gewoon het ja, als jullie de discussie stilvalt, dan kunnen wij nog een paar vragen stellen. Maar wij zullen dus
ik heb voor iedereen nog meer dan
tijd in de gaten houden. En dus straks dan hier de volgende is voorbereiden. Maar dan we beginnen om 5:30. Ja, als jullie de discussie stilvalt, dan kunnen wij nog een paar vragen stellen. Maar wij zullen dus
ik heb voor iedereen nog meer dan
Okay, if everyone can hear me, we can start the first discussion. We're going to start with the creativity of Lexus detectors and iXense, starting with Lexus. First, we'll play the pitch, then it will be 10 minutes of the discussion, chaired by our jury chair, Wietse Watman. He will give the word to other jury members, and then there will be a discussion of 10 minutes. So the pitch can be played right now.
My name is Felipe and I'm part of the LSS team, and I'm here today to tell you all about our biosensor. So imagine that you want to measure the quantity of valproic acid in your blood. You have to go to a clinic, you take a blood test, and in a couple of days you'll get the results. With our sensor, however, the time of the analysis is much shorter and you can do this test anywhere you are. But how does it work exactly? One fundamental component of our idea was to bring SAT3, which is already used as a therapeutic target in cancer research, into the study of epilepsy. We also found out that VPA binds directly with HPAC3, and this feature is what makes it possible for our biosensor to be able to measure only the fraction of VPA that is free, which means it's not connected with albumin. Then HPAC3 will form a complex together with SAT3. And this complex will inhibit the phosphorylation of the STAT3. As a result of this highly specific inhibition, STAT3 does not suffer dimerization. Moreover, we also looked at the action of ferrocin, which will have a key role in generating a signal that we can then read. Due to COVID-19 restrictions, and because we wanted to support our idea, we had to use a biochemical software, Biomolecule. The software enabled us to look at specific binding sites of interest, such as the VPA with the HPAC3, as well as the ferrocin with the STAT3. And with our simulations, we came to the conclusion that the binding sites that we were interested in were actually highly specific. Now that we've gone through the molecular pathways for the detection of the VPA, how exactly are we obtaining a signal? Well, for this purpose, we use the screen-printed gold electrodes on which the ferrocin reaction will take place, and the SensUs Smart device, which is a potential start sold by PalmSense. And then how does the mechanism exactly work? Well, let's keep it simple. If you have VPA in your sample, the dimerization of the STAT3 is going to be inhibited, as we've gone through, and then a ferrocin reaction will take place. If this reaction takes place, electrons are transferred and the current is created. And so you have a current. How is this current converted into a concentration quantity, let's say? Well, we use a square wave voltammetry. And in this picture, you can see the oxidation peak of ferrocin, and you have to see that if there is more VPA in our sample, this oxidation peak will also increase. In order for us to obtain these specific values, we have to go over these experiments a considerable amount of times so that a calibration curve can be obtained. Coupled to our biosensor, we have a fully personalized app that our patients can use to self-monitor them. In our app, they can create their own personal profile where they basically keep a record of their latest exam results, and they can create their seizure diary to take notes of symptoms, etc. On top of that, they'll be in constant touch with their neurologist who also have access to the platform. If they want to, they can also talk with others, with other patients that are using our platform and share their experience. I hope you enjoyed this presentation, and see you soon.
Okay, thank you very much for your nice presentation. Can everybody hear me?
Yes.
Yes.
Okay, that's good. That's good. Yeah. I would like to give my other jury members, Jasmine and Hassan, a chance to ask the first questions, and if they are done, I will have a few for you as well. Jasmina, will you start off?
Yes. Let me see. Sorry. Okay. Well, again, congratulations for your presentation and for your work. Well, you have given a lot of details, but I still would like to know a little bit more on, for instance, have you thought about other species that could also oxidize during the readout of your process, that could interfere in some way besides that could be present in plasma or in blood?
We thought about that, and we talked to, we had a specific professor that helps us in this area, Professor Anna Vienne, as she is very deep in her research area, is very deep in electrochemical biosensors. And she told us that since the only problem that we might have had was related to oxygen, but since that could interfere with the oxidation points and hence interfere with our readouts, but since we have, like Felipe told us in our pitch, a very specific reaction and binding points between the enzymes, and we actually are using two different enzymes. STAT3 is the main one, but valproate actually binds to HDAC3, and it's like a complex reaction that goes into a chain. So since we divided that in different points, and they're all actually very specific, we don't have that problem with other interferences. Of course, we didn't get the possibility to test it, which is unfortunate due to COVID-19, but our simulations tell us that we should have no problem.
Okay, good. Okay, thank you. Well, if you can proceed with other jury members,
Okay, thank you. Thank you very much. Hassan, may I give you the word? Hassan there?
We believe that Hassan isn't in the call yet, so I think maybe proceed with other questions.
Then I will have a question for you. One of the big problems for valproate is that it has a rather narrow therapeutic range, and has to be, I think, if I remember well by heart, somewhere between 10 and 50 microgram per milliliter. From your beautiful work, but also from the very complex chemistry, you gave absolutely no clue where your sensitivity is and in which range you have sufficient accuracy for your determination. Do you have any idea?
That actually is on the main paper we use as reference. I can try to go get it because I don't know the value by heart. But since our method is very similar to theirs and their range is in the same as the ones we were provided for the SensUs competition, we thought that we might be okay since the basic method of detection is the same and their range is in accordance to the ones that were asked for us. I can try to find it. But...
No, it's okay. If you checked it, then I believe you. We don't have to go time for the details. And how about your accuracy? I mean, this is very complex chemistry. How reproducible is it? Is this something where you will get the same number every day?
Again, we hope so. Our simulations tell us that probably, but we would have to test it. And unfortunately, we didn't have that opportunity.
You have any idea where the most important sources of error would be?
The most important sources of error? Well, our reaction is dependent on different proteins. And if something reacts to one of them, or if they don't bind the way they're supposed to, if we have a slight error in the concentration of the proteins that we apply, a lot of things could go wrong.
But if you go even to the patient, right? All starts with the patient. And you know what the biggest problem for epilepsy patients is and their drugs control? You have any clue? What's the main problem for the epilepsy patients?
Regarding?
In their medication.
What you're talking about, side effects or?
I'd say probably
in the maintenance of epileptic patients. The important issue, they simply forget their medicine.
Oh.
Okay. Yeah.
I would say maybe
the other problem is that you have to adjusting a concentration takes a very long time.
Yeah, it does.
So I wonder whether measuring daily your free valporate will be of any help.
But that's also like kind of the point of the fact that we chose a particular group inside epileptic patients. We chose children particularly because of the fact that their concentration range changes a lot because of their weight and height.
But then do you trust the parents to adjust the concentrations? If that's
No. Since we are connected, since our sensor can be connected to our app and the app is connected to neurologists, what happens is when we have a concentration that is not in the limit of their height or weight, the neurologist will be alarmed to set an appointment to try to readjust the medicine.
Yeah. And do you expect any problems in the controlling authorities if you work with children? Because that's in the pharmacology area, that's a very sensitive issue, right?
Yeah.
And in fact, if you go into the details, very little is known about pharmacology in children. So
yeah, there's no clinical trials.
Yeah, that's something. But just to add something about this, you were saying if it made sense for people to measure their valproic acid daily, like the concentration. But actually, from the feedback that we got from doctors, we tried to interview several doctors about this, and we realized that maybe what made sense was, for example, one measurement per month, and then for them to keep on our platform a track of their symptoms and how they're feeling, because measuring every day wouldn't make so much sense because even in children their weight doesn't change that often. So yeah, so our goal was not to measure daily. It was to measure. Yeah, it was
and the
to measure.
weight is, of course, only one of the factors, right? There are many more metabolic issues.
Yeah,
weight is what we see, and that's what we see if they grow. But as you might remember, a lot changes when you're a child. It's not only your weight.
That's true, and valproate is probably not the only medication they are taking as well. So that's also a complication on that.
And you also expect no interference with other medications?
That is also mentioned in the BASED paper, because they do try to see with other medicines, and the thing is, the proteins that we are used, one of them is a, how do you say it in English, a nuclear protein. So the fact not a lot of pharmaceuticals can interfere with it. It's a very stable protein. The same thing with HDAC3. So there are not a lot of studies that mention interference with other drugs because they're very stable proteins.
Well, we use the same voltammetric method to measure dopamine, for example, which is also present in the blood, etc., etc. So it would be nice if you could test and see how all these issues work out. I think in view of the time, we might have to go to the next presentation. Is that so, SensUs?
Yeah, that's totally all right. I also would like to mention Hassan Asazi, the other jury, is also in the call right now. So you can give him the word as well. But we'll continue with the next presentation.
Hassan is now there?
Yeah, he's now here.
Okay, good.
So we'll continue with detectors.
Thank you.
Hello, I apologize for the delay in showing up, but I have some internet issues.
Okay. Hi, my name is Kalami, and I represent the Danish Sensors team, Detectors. This is our creativity pitch. Our concept is based on the giant magnetoresistance effect. Essentially, this is a magnetic sensor, but more on that later. The sensor surface is functionalized using antibodies specific to valproate. When a blood sample is added, the valproate will bind to these antibodies and give a representation of the valproate in the sample. A secondary antibody bound to paramagnetic nanoparticles is then added to the solution, and excess is washed out. This results in a sort of sandwich where we have the nanoparticles bound to sensor surface via the valproate. When inducing a magnetic field, the paramagnetic particles will respond with a field of their own, partially negating the induced magnetic field at the sensor surface. This leads us to be able to count the particles that are bound to the surface. The transduction method magnetoresistance is a quantum mechanical phenomenon. Electron spin leads to electron scattering in the presence of an external magnetic field opposite to the electron spin. This essentially means that in a ferromagnetic material, electrical resistance is dependent on magnetization direction. Layering these materials, this effect is increased greatly, thus the giant magnetoresistance. It is seen that the more parallel the magnetization of the layers, the lower resistance of the materials is. Since resistance to an electron is just electrical resistance, well, the GMR sensor can be modeled as a variable resistor and easily be used in circuits. This leads to the number of valproate molecules being able to be read directly from an output voltage. In order for this method to be able to detect valproate, 3 requirements need to be met. First, valproate needs to have 2 binding sites. Secondly, we need to be able to functionalize the surface by immobilizing the primary antibodies. And third, we need to be able to bind the secondary antibody to the magnetic nanoparticles. Now, valproate is a very small molecule, but it has been shown in the literature that it has 2 binding sites. To test if we could bind antibodies to these, we would propose 2 plans. First, we would like to use a sandwich ELISA to see if you can bind these 2 antibodies. And if not, we propose to use bovine serum albumin as one of the antibodies and test this with a competitive assay. We have not been able to do this due to COVID-19. Now, the surface functionalization should be doable since a lot of consumer electronics are layered with a silica layer at the top. Now, there's a very well-known chemical for functionalizing silica surfaces called APTES. The nanoparticles are covered with gold, meaning we can use EPS and NHS to functionalize the surface. Due to coronavirus, we were not able to create a working prototype. However, we were able to develop the concept, create a testing platform and a calibration platform, and the interface for the device itself. Thanks for your attention.
Okay, thank you very much.
Thank you.
Hassan, since you're there, will you kick off the discussion? Let me see.
You should unmute yourself, Hassan.
Okay. Can you tell me, first of all, thank you very much for your presentation. Can you tell me how you will be able to differentiate between free and bound valproic acid?
Caroline?
Yeah, it's supposed to, it depends on whether our ELISA works. We want to test the interface first with an ELISA, and if this method doesn't work, we want to use a competitive interface. The competitive one will be like with fluorescence, where we use a BSA and VPA conjugated on top. So depending on if we use like the competitive, we will only be able to measure how much free valproate there is, and the ELISA, that's free as well.
Okay.
Yeah.
All right. Okay. Thank you.
You're done, Hassan?
Yes.
Okay, then Yasmina, can you continue?
Yes. The clinical range of valproic acid is quite short. Do you have any idea about the change in magnetic field caused by the number of particles that you are going to have in that range, and if that could interfere in some other magnetic fields that we may have around the sensor?
So so definitely, like the magnetic field will of course depend on the size of the magnetic particles that we bind to the surface, right? But I think we calculated that. Oh, what did we calculate? Like, it would induce a field of around 10 to the minus 7th Tesla or 8th Tesla, something like that, per molecule nanoparticle bound, which in GMR sensing is measurable. And sorry, what was your question again?
No, I mean, and do you think that is there any equipment or anything that could interfere with that amount of?
Definitely.
Yeah.
Yes. Like if you put your phone next to it, it will probably change
exactly.
it as well.
And if that happens, how can you isolate that? Or did you have anything in mind in this?
Yeah.
Well, like if you put like the magnets on your speaker on your phone, for example, if they're close enough, it would induce a massive spike. So it could report it as a false measurement.
But we do have a method for negating this, in that we have built in a sort of calibration measure in the system, where we when we induce the known magnetic field, well, we know the field strength, so we can kind of normalize the field around, like the measured field from the sensor to this known field strength.
Okay.
So before doing a measurement, you would do this calibration, which should be really short, and then it would do the measurement. Okay.
And then there would be kind of some requirement for whoever is using the device, so they know that there's certain devices that cannot be near the device or something like this.
Sure. I mean, probably the main requirement would be not to, like while measuring, not to change the position or put something near it, right? But if we could, like, since this measurement is pretty fast, because every time a particle binds to the surface, we can see it instantly. It should not be a long time that you would have to abstain from moving the device.
Okay. Well, thank you for your answers and for your nice presentation. I don't have more questions.
Okay, thank you. Then it's my turn. It already came to the floor, but the antibody type of measurements, ELISA type of measurements, they are extremely sensitive. So that's a great issue. But at least in our hands, and I think in many people's hands, quantification is not an easy issue there. And you mentioned already a few times that you have to calibrate something at some point. Do you have to recalibrate every time you measure, or every time you take a new sensor, or you take the next sensor, and how is the calibration done? Against what do you calibrate?
Well, first we do a sort of characterization, like in the beginning when we do create the device. We do a sort of basic calibration of the sensor, where we measure its response to an induced magnetic field, right? Because this response is nonlinear. So it's not like every time there's a particle, it gives a linear jump up.
No, but that's why you have computers, right?
Yeah, exactly. So we have this sort of curve programmed into like the device itself for each sensor we have, and then when doing a measurement, we would do this calibration where we just induce a magnetic field to sort of normalize the external fields.
And you think the average epilepsy patient or its parents will be capable of doing that?
Well, the basic, like the first calibration will be done from our part. Like it will not be anything that they would have to do.
Yeah.
And the second calibration we would just do with a press of a button, right? Because we can the electronics used to do this measuring is, well, that's just making this magnetic field. And you can use the same magnetic field that you induce to measure to calibrate. So it's just pressing a button and then it would do it on its own.
So we do it by electromagnet in principle.
And most likely your software then gives a number, right? The VPA concentration.
Absolutely.
And what happens then?
Well, Our
one remark in your document that I found a bit scary, that one of the major profitable things of this whole approach will be that you can avoid the doctor, right?
Oh yeah, I think maybe that's
the visits to the doctor.
Right, I get it. So I think maybe
think about liability. If your instruction says raise your valve rate because the determination was low, then the people dies.
Absolutely. I think maybe we didn't specify that clearly enough, but I think the way we would like to do it is that you could avoid going to the doctor, but the device would send the measurements to the doctor, and then he would be able to call you and say, okay, you should do like this and this. So it's.
And what did you ask the doctors what they think about it?
I think Yusuf got in contact with one doctor.
Yes.
But it was.
Remember exactly.
Right. It's a conceptual on a conceptual level.
I mean, I have some experience with epilepsy, and I think it sounds like an easy disease, but interpreting how well the patient is doing is absolutely not an easy thing. And I wonder whether doctors would simply take your number and then adjust people's medication, or whether they really want to see the patient.
Well, hopefully not. Hopefully it's a holistic view. Like hopefully they know the patient and they know their responses before they give them this device, right? So so it's a supplement to the.
Yeah, but you could say that every 5 times after 5 times of a concentration, it would be good to look at the patients and doctor test that they at least are still alive, right?
Yeah, that would be great.
Well, the previous questions I had also holds for you, of course, but you could not measure it, so you have no idea in the sensitivity range what your accuracy is there, right?
Unfortunately.
How reproducible would it be? I mean, because if it's 10%, it would be okay, but the small window might be a complicating issue.
Yeah, absolutely.
Right. I mean, I don't remember, but you can probably explain me. The patient has to simply put a drop of blood into the machine, or does he have to do more things with it before?
No, a drop of blood, and then we would hopefully do some simple microfluidics to be able to deliver a certain volume of blood to the device, so we can measure the concentration, right?
And does he have to be very careful in cleaning, for example, the machine or the device? For the next day?
So the way we envision it is that we have a cartridge that you can insert into the device, and this cartridge is, it's disposable, right?
Single use.
Yeah.
Okay.
Also, this cartridge would mean that if you were able to do this method, you might be able to do it with other medications as well. So you might be able to have different cartridges for different medications.
What kind of other medication would you like to combine? I mean, how horrible are these patients?
I don't know, but you know that
Alzheimer, Parkinson, and epilepsy all at the same time.
No, but in case of like a multi-therapeutic, like where they have multiple drugs for epilepsy, you might be able to measure all of them.
Yeah, did you discuss that with the doctor? Because that's the epilepsy nightmare, right?
Yeah. Exactly.
It takes about half a year to really get a patient stable on a certain medication. There are what they call the honeymoon weeks, where every drug always works, and then very often the efficacy goes down. So you have really to wait for a couple of months before you really know whether it works. Now, if you think about using 8 medicines and you have to test them, taking half a year for each medicine,
that's a nightmare.
The patient is dead by the time that you
absolutely
any idea what would be the right concentration. So
that's
sounds
actually
very nice, but the treatment will be extremely difficult. But it would be nice if you could give it a go.
Yeah, I mean, it would be helpful in these first stages, right?
You think that the other epilepsy medicines there are, like phenytoin or Keppra or any of the other things, could also use the same methods that you propose? Is it universal?
I have to say that we have to close off the discussion soon. So maybe a last few words.
Right.
It would depend on size mostly.
Exactly.
But That was it.
But we'd have to test it.
Yes.
We wait for the company.
Yes.
Thank you.
Thank you.
Okay.
Thank you for your time.
Great discussion. Then I think we're going to continue with the pitch of iSense. So it will start in a second.
Hello, greetings to everyone. We are the team iSense from Eindhoven University, Germany. I would like to start our sensor development for valve rate measurement with technological novelty. We are using metal organic framework or MOF for sensing valve rate. MOF is a solid crystalline structure with metallic centers bonded to organic linkers. Here, MOF we have chosen is terbium-based MOF. It is deposited over the gold interdigitated electrodes. The next one is our sensing strategy. The sensing strategy we are using is electrochemical impedance spectroscopy. It requires less equipment compared to other techniques and it can be made portable. This makes our electrical readout easier and also the sample volume we are using is very small. Our fabrication technique is simple and reliable and therefore we can manufacture this in large quantities. Then I'm moving to molecular recognition part. As I said already, MOF is deposited in gold interdigitated electrodes. MOF provides large surface area with considerably good mechanical and chemical stability. Due to the surface functionalization, measurements based on luminescence, field effect, and impedance is possible. In our case, valve rate molecule size agrees with 4 dimension of MOF. Hence, valve rate can be identified by surface functionalization.
Physical transduction. Measurements are done using electrical impedance spectroscopy with a 3-electrode setup. Here, one of the finger pairs, the working electrode, is coated with MOF, while the other finger pair is left uncoated. The reference electrode is a commercial miniature AGCL reference electrode supplied through an inlet. Upon contact with the analyte, the impedance changes over time of the working electrode, which is registered at a specific frequency. To get reliable measurements, both the impedance after a specific time frame as well as the slope of the impedance change over time is considered. So through data statistics, a correlation between the amount of VPA and impedance change is achieved. The Cartridge technology. Sensor consists of a 7x7 mm dies with a PDMS microfluidic system on top of the chip. These dies are glued on a chip carrier and electrically connected with bonding wires, which are passivated using PDMS. The cartridge consists of these chips cameras, including the microfluidics system and the chips, and can be connected to the device by insertion into a chip socket. Reader instrument and user interaction. Readout of the impedance measurement is done by an MSTAT Pico, which is connected to the 16 IDE working electrodes via multiplexes, since the MSTAT Pico accounts only for a pair of electrodes. All channels share common counter and reference electrode, which are directly connected to the MSTAT Pico. The MSTAT Pico communication is read out with the microcontroller via UART, so the microcontroller can calculate the VP concentration using a calibration curve. The calculated concentration is communicated to the user using a display. Additionally, raw data and a log of results are stored in integrated memory. A cartridge change can be easily done by the user by removing the cartridge from the spring-loaded socket and inserting the new cartridge. Our technological feasibility. The sensor uses a microscale gold interdigitated electrodes on a glass substrate, fabricated using standard clean room lithography technological process. Therefore, fabrication is cheap, allowing mass production and very high reproducibility. These IDEs allow precise electrochemical impedance spectroscopy as well as functionalization of every single electrode separately as done here using metal organic framework to enhance the impedance readout. This allows different MOF layer characterization on a single chip. The electrochemical MOF deposition process has shown high reliability and reproducibility and good mechanical stability. Thus, as an overall matching criteria, iSense can produce a reliable and an efficient biosensor for valve rate measurement for the millions of people who need it.
Okay, thank you. Well, thank you very much for your presentation. May I ask Hassan to start a discussion?
Sure. Hello. Thanks very much for your presentation again. And I was just wondering, why did you use terbium, in particular, terbium TBC MOF metal organic framework? Can you tell me a little bit about the reasoning behind using terbium TBC MOF in your sensor?
I guess the practical reason was that our supervisor has a cooperation with a chemist who does research in that area, and it was just that we had the samples available of this particular MOF and it just happened to be.
I saw this in the report, but I was hoping that you can tell me a little bit more, or at least discuss with that chemist what did he use terbium as as a rare earth element in this setup. I think it's important to understand the selection, because this, I believe, will have to do with the ability of this layer to properly conduct better signals. Anyway, okay. Again, the question again will be, how do you plan to differentiate between free and bound valproate?
So I think, well, the idea was that depending on like the parameters and electrical characteristics during deposition, we would variate pore size, and if the valproate is bound to like large proteins like albumin, then it would not able to get as close to the electrode.
But the equilibrium between free and bound, you have some valproic acid bound to albumin and other is free. So when free start binding, maybe some of the bound will let go of the albumin and start become free, and this will not give you necessarily a correct signal with the actual amount of free valproic acid in the sand.
Oh, so you mean that there's like kind of a dynamic equilibrium that changes
during
the...
exactly. That's
okay.
That I'm concerned about, yeah.
Okay. I don't know. Does anyone else? Because I don't think we discussed it like that.
Okay.
Because I think we just would just assume that it would be static. So we would have like after administration of the drug, we would have a certain free fraction, and that would not change during the measurement. So.
Okay. All right. Okay. Great. Well, thank you very much, and good luck to you.
Thanks.
Thank you.
Okay, Yasmin, can you continue? Yasmina?
Yes. Picking up this coating with the metal organic framework that you are using, which is one of the novelties of your sensor. Could you tell us a little bit more on which advantages that kind of functionalization provides compared to other more conventional, such as using antibodies or other enzymes or things like that? For instance, let's say in terms of stability of the sensor, do you think it's going to be easier to distribute because it will last longer, or did you consider that?
So one thing we were thinking that it's probably easier to deposit. So you just have the solution that we had, and then like it's applied to the electrodes, and then we run the cyclic voltammetry. So deposition should be easier. The previous year's team, they had like a novel way to deposit the antibodies on a I think was it PDOT? I'm not quite sure. On a polymer substrate, and the problem they had was that the orientation of the antibodies was not always ideal, and we thought we could like circumvent this. Also, antibody handling usually has to be at certain temperatures, has to be stored at certain temperatures, and antibodies are quite expensive. So we thought it would be easier if we could do it without antibodies. But I guess like in testing, we didn't really get to the stage to see if just the MOF suffices, if the selectivity is enough. So just in case, we also ordered antibodies, we're thinking about employing it, but due to different problems we encountered, we didn't get to that stage to also test.
Okay. And again, once you have your coated electrode and you have used it, which is the procedure? So did you thought about what is going to happen? So it's a single-use cartridge. It can be reused for a certain amount of times. It can be washed or not.
So we didn't try it, but we were assuming that it would not be possible to reuse the cartridge. The IDE devices we had, they have 16 different electrodes, so theoretically it could be thought about like kind of compartmentalizing it, so like the different electrodes could be used if like maybe like a cover is being used. So during one measurement. Not all electrodes are coated actually with a sample, but yeah, we didn't test it, but we were just assuming that would be a single-use cartridge.
Okay, good. Thank you. Thanks for your presentation and your discussion. I don't have more questions right now.
Okay, then I may continue. Thank you for this really original idea. There is one thing that I would like you to explain me a little bit more in detail, because I didn't grasp it, I think. What is the basis for the selectivity for VPA in your method? Is that size of the VPA molecule, or just only size? Or is charge also important?
Anyone else? Because
yeah, it was size only.
It's only size?
Yes.
Actually, we have seen research papers that said that this molecule valproate could actually be detected with the help of MOF. So it's basically the size that is considered.
Would your measurement be affected by pH of the blood, for example?
This would we have to test in further measurements, but right now we're not sure, of course.
I don't think it will be affected with the pH of the blood, because we had referred previous papers, like done a literature survey on MOF, and there was a paper that said that using with blood also MOFs are used to detect valproate. But it wasn't like on the chip detection, but it was actually done.
Okay, and the same holds for ionic strengths, for example. Is that a relevant issue? But these are all things that are quite complicated in blood, right? I mean, it's not saline.
So what you're aiming at with the question referring to pHs, that like arguably the speciation of the valproate might change due to pH, like so like some of it, like sometimes it would be charged, sometimes it would not be charged, like the different fractions change.
Could well, I wouldn't be surprised. Yeah.
Yeah. Unfortunately, we don't have very many measurements to kind of like buttress the theory, because
I had to kindle your thoughts a little bit.
Some problems with the passivation of the chips, and then like some delivery of the valproic acid, which did not arrive until shortly before the end of the deadline. But
the same holds for all the effects that influence the equilibrium between bound and free, of course. And there is, for the epilepsy part, an even more important issue, which of course you were not supposed to deal with too much, but it may come up if you put it in practice, namely that the free concentration in blood is of course relevant, but 10 times more important is how much of that free concentration passes the blood-brain barrier and ends up in the brain, where it actually has to work, right? And I'm not so sure, people do not know exactly how the blood concentration relates to the brain concentration. So you might finally discover that although the concentration in the blood is very stable, the control over epilepsy is not, or the other way around. So
but I mean, like that's kind of like the next step, right?
Yeah, yeah.
Like we can measure the blood concentration, and then like that's up to further studies and doctors. And
your easy solution would simply put your sensor in the brain, of course, but
yeah.
Not so much for the patient, right?
Yeah.
But what I got from your document, at least, you're one of the very few teams that at least gives a try at the sensitivity in your figure 2.2, if you remember that. You have dots where you at least relate your signal to the actual free concentration of valproate. What I wonder is if you look at the relevant range, which is only very small lower part of your graph, I mean, there's only a single point there. Do you have already any idea about the reproducibility and the sensitivity in that range? Is that a good range for the sensor? Because I think your graph in the figure 2.2 goes up to 500 micrograms.
I'm so sorry, but I will have to cut off this discussion given the fact that in a few minutes the translational potential discussions will start. So if you want to get in contact, I think you have each other's contacts right now. And then I would like to thank everybody for participating, especially the jury. And yeah, then we'll start calling in the next round. Thank you so much.
Thank you.
Thank you.
Thank you.
Thank you.
Darius, are you present? You will have to unmute your mic if you want to say anything, but I think you're present, so that's nice.
Hello.
Hey, Stuart.
It's not Stuart, this is Hassan.
Hi, Hassan. How are you?
Oh, Hassan.
Are you done for now? Or
yeah, the discussion is closed, so you can leave this call.
Thank
and
you
we
very
will
much.
start. Thank you so much.
I have a question.
Bye-bye.
Yes, Claire?
So do I need to stay in the session?
Yeah, you're supposed to be in the session.
Okay.
So in the next...
You're not yet in the session, right?
Sorry, you're?
The startups are not yet in the session.
The startups? The teams, you mean?
Yeah?
Yeah, and they are now joining the session slowly and will start in a few minutes.
Okay.
Right.
Can I chat or something?
Fleur.
Stuart?
Hello. How are we going to do timekeeping in this session?
I will do the timekeeping. So my colleague will play the video, and then you have for 4 minutes. Then we have 10 minutes of discussion, and then I will tell you when to move on to the next pitch.
Brilliant. Thank you.
Okay, and are we now like in a closed group, or are the teams already joining this session with you?
The teams have already joined, but they're all muted and with camera off, so they will unmute themselves when it's their turn.
So they can hear us already.
Yeah.
Okay.
They can hear us. I think we can start with the first presentation of Sense Glasgow. So we'll just show the pitch, and afterwards there are 10 minutes of questioning. So,
okay, perfect.
Hello and welcome all to Sense Glasgow presentation of its biosensor translational potential. Please feel free to ask any questions at the end of the pitch. So I'm going to guide you through the 3 key parts we approach to develop our biosensor model, the first one being our customers. We thought about 3 main ones: patients, doctors, and researchers. Each one of them, in fact, has many reasons to buy our biosensor, which add value to our product as an alternative test that, apart from being cheaper, is more convenient and faster. The idea is that doctors and researchers recommend our product to patients, while our distributor key partner convince the professionals through scientific presentation and marketing strategies. And that customers learn how to use the product through manuals, reference from other people, or medical institutions. Using this information, we can start building up what our biosensor business model canvas will be. And we can move on to the next section of this model, that is the product itself, which is built through a first stage of development, followed by a manufacturing stage and a marketing one. Each one of them requires their own key resources and professionals to handle these activities, which will be supported by our key partners in terms of, for instance, distribution strategies, marketing, and supplies, among others. This completes the second stage of the business model canvas, and we can move on to the next and last section, the cost. We consider a cost-based approach, where we add up all the expenses needed to build the biosensor, and a market-based approach, where we see the range of prices of the biosensors in the market. We end up setting a possible device cost of £25 and £50 if a pack of 3 is bought. The revenues will come not only from distributing the product to sales business or putting up in online medical markets, but also by licensing the product. So what's the challenge? The market size. We need to convince our licensor that the product we are providing will bring both companies significant benefits. And focusing only on patients on the valproate and its doctors could give a maximum of 48,000 tests per year, which is definitely not enough. This number is too ideal, and there are many adversities it needs to face, such as denied advice or doctor suggestions. Reason why our team found 3 other ways to expand our market. Starting by a design that can detect more than 30 other medicine concentrations apart from valproate, followed by aiming to a global market where buying this biosensor becomes more effective and time-saving than in their own country. And finally, offering this biosensor as a companion of a genetic study, where at-risk patients may also consider valproate as a treatment, even with a high risk of hepatotoxicity. This market expansion allows the market size to grow to a minimum of 1 million possible customers, which increase the chances of obtaining profits from it. This information finishes our business model canvas and our planning. Thanks so much, and do you have any questions?
Okay, so if please the jury members, Claire Stewart and Darius, could put on the camera. I already said some did. And then we can start the discussion, and also the team members. So let me see. Marie Carmen should be in the call.
Hi everyone. So this is Sense Glasgow, right?
Yes.
Yeah. Okay. Thank you for your clear presentation. It was a different pitch actually than the pitch that I saw before. I should introduce myself also shortly. I'm Claire Visser. I'm working for Yes Delft, incubator in Delft, partner of EIT, and responsible for the MedTech focus area. I had a question. How are the patients and other stakeholders considered in your business model?
What do you mean exactly? Do you mean how are the patients going to use the biosensor? How are we going to attract patients with the biosensor?
How did you validate your stakeholders? Like, did you execute it like customer interviews, actually talking to patients, payers, providers?
So we talk to doctors first, and then from there we survey patients, honestly. So we went to a hospital and handing papers, and then they give us back, or survey online. With already like predetermined questions. That's how pretty much we approach the patients, because due to the pandemic, we couldn't contact them so closely. So we were relying on the people that were passing by in front of the hospital. And then besides, our team is like really international. So our data is also pretty international and pretty difficult to get like focused on a place.
And what was your main learning from the patients?
Or main learning, I guess in terms of their feelings, I guess they are pretty
not scared, but they rely too much on the doctor's opinion. That's what we learned. Like they rely on their doctor's opinion or their supports on societies for epilepsy. They pretty much are a community that build their opinions together and are closely related. So if we contact one, we can get access to the other one.
And this learning from talking to the patients, how did you incorporate it in your business plan?
So we incorporated in the market size. Like we thought this was the main way to get the market size because there is no much data available online. And I think for the design as well, we consider the patient's benefit. I think that's all.
Okay, thank you for your clear answer. And I was wondering how you look at your competitive advantages. How do you position yourself in the market to existing similar existing innovations or like main practices now, or also your competition?
So we only thought about other biosensors in the market. We didn't find any other valproate biosensors. And we consider them in terms of price. So in a range of which price should our biosensor be, that to get the willingness to pay of the customer, basically. And we also consider that if our technology is reliable enough as theirs, it could get accurate results and it's fast and convenient for the patients as well. So we saw there are many people that need to go to the hospital to get tested. So the people that live in the countryside needs to travel all the way. And some companies offer the test only in the hospital. So our biosensor is sold on a pharmaceutical way, or online or in the online market. So we can, so they can get it either for delivery or from the pharmacy, in local pharmacy.
Okay, and I have a last question, if I may, and that is, I was wondering, what do you consider now as your riskiest business assumption?
My riskiest business assumption?
So to continue in the future, what is an assumption you made that you think is actually the riskiest assumption and you need to tackle as soon as possible?
That's a good question. I think the advice from the government laws, probably.
Okay.
Our base is in Glasgow, right? So it's in UK, and our biosensor is pretty much based on valproate, at first instance, and then we move on to the other drugs. And the nice advice that is the entity in the UK regulating this sort of stuff. Doesn't recommend the continuous monitoring of the valproate for some sickness such as bipolar disorder. So it's pretty much we need to see if in other governments or even in the UK government we still can is it recommended or not to monitor drugs continuously or not. I think that's the main risk, because if it's not recommended, the people won't follow us.
Fair enough. Thank you very much.
Hi, Mary Carmen. I'm Stuart Lowe, consultant at TTP. I have a question as well, just about how are you looking to raise investment in your company? What investment model are you considering?
I guess through presentations and pitches to companies to look for investors. I didn't think of anything else as a marketing strategy to sell our idea. We thought
is this to the VCs, or kind of who are you going to approach to invest in you?
Who am I going to approach? Good question. So I think my lack of experience to know exactly which company I'm going to approach.
But you're looking to kind of sell to an existing company, or are you looking for kind of VCs, investors, or I suppose the question is how do you see your company growing? And is it like an exit as well?
I got your question. So I think we are thinking of an international market, right? So we think of licensing the technology to companies that are widely recognized, like around the world. The thing is, as I spoke to doctors, I realized that they rely mostly on the companies that they have been working on for a long time, right? So we want to licensing the product to existing companies that actually like may be more like reliable for other countries. I don't know if that answers your question. I don't have any specific name.
No, that's fine. Just kind of that's a good model. Do you know how thoroughly you will need to prove the technology before you can sell a license?
How long? I don't know.
Or so what are they buying a license to? In effect.
The biosensor itself, I think, like so for example, the biosensor may have many uses, right? So they may use it to test for clinical trials, for example. They may use it to accelerate the process and try to see if their new drug is actually working or not on patients. They may use it to I don't know. I'm thinking right now.
So
they may use it to sell it also to the patients, like as a control for them. And also, as I was saying in the pitch, like the companion of a genetic studies, so they can sell it to doctors, so they can ask the patients to use it and determine if the valproate is an option for them, like considering that like their genetics perspective.
Okay. I think we're on the maximum of our time, so we'll have to continue with the next pitch. I want to thank Sans Glasgow for the nice conversation. And we're now going to start the pitch of EpiSense.
Okay, looking forward.
You.
Hello, and welcome to the EpiSense keynote of August 2020. Today we will unravel our business plan for the future and hope to have you on board on our journey. If you saw a 1-minute pitch, then you would realize that the life of a person with active epilepsy is not easy. The medication they take, valproate, has a different dosage effect on every individual. Because of that, the average epileptic person undergoes nowadays an avalanche of time-expensive treatments in their quest to live with epilepsy. The reality is that around 5 million people in Europe suffer from epilepsy, resulting in a direct and indirect cost of 6,000 euros per patient per year. Moreover, when this neurologic disorder is accompanied by kidney impairment, hepatic disease, or hypoalbuminemia, there is no effective way to determine their serum levels. Lastly, in emergency situations such as status epilepticus, valproate levels need to be determined very fast to ensure the right dose administered. So in essence, there is a need for a precise, cost-effective, and fast therapeutic drug monitoring device for the measurement of free valproate at the point of care. This is what we offer, with the EpiSense device and cartridge. Now, what are the main benefits for our stakeholders? For the patients, like mentioned before, a far more effective treatment, and finally a good solution for patients with underlying conditions and those in emergency situations. For the neurologists and hospitals, there are economic and time benefits, as there will be a lot more patients reached while the amount of interactions decrease. And for our pharmaceutic partner, Mylan, we offer them a higher market share of their valproate drug in exchange for distribution of our devices and cartridges. Together, we will expand all over Europe and beyond. Let's talk about the future. This year, we will start the development phase, followed up by a clinical study the next year with key opinion leader from UZ Leuven. This will allow us to validate our device proof of concept, and market reach. Next up is the start of bringing our product to the market with our distribution partner, Mylan. First to the 4 centers of refractive epilepsy in Belgium in 2023, followed up by an expansion to the big hospitals and neurologist clinics in the Benelux. We aim to be well established by 2025 with a market share of 11.5%. The same approach will be followed to succeed in our expansion in Europe. The aim is to sell devices to the epilepsy centers in the capitals and big cities of the countries in the European Economic Area, plus the Balkan countries and Switzerland. After 2027, our future goals are to expand our market further in North America, expansion to other anti-epileptic drugs, to create and distribute the at-home product. This requires a ton of research and solving of legal and privacy issues. And finally, to create and distribute an altered, optimized product for low-income countries, where 80% of all people with epilepsy reside. Here you can see our commercial plan with all of our key partners regarding funding, production, market expansion, distribution, and us doing further research and development. Next up, our financial plan. The big takeaways are that we start out with 1.1 million seed capital. We plan to be break-even after 6 years and reach 1.3% of operate users by the end of 2027. We hope you enjoyed our presentation and look forward to have you on board. Thank you for listening.
Okay, thank you, Team EpiSense, for your presentation. I'd like to throw the floor to any of the other jury members first. If not, I have a question about your relationship with Mylan. The business plan is quite dependent on them as a distributor. Can you tell me a little bit more about how that relationship is going to work in practice?
Hello. So yeah, Mylan is our distribution partner. They are like a big pharmaceutical company. They are also a player in valproate medication, but they are like a very small player. They have some portfolio of players of medication, but they're not as big as the biggest player in Europe, for example, Sanofi. So what we offer is that we include their medication, so their valproate drug, with our product, our EpiSense cartridges and device. So in that way, all of the patients that use our device, and I mean the neurologists and the patients that use our device, they will use their valproate drug. And the increase in revenue that they will get from that, we calculated, will be far more than enough than the cost that they will have to distribute our product. There's always a possibility they might ask for a cut of our cartridges. Like we know it's around 30%, but we are very confident that the raise in revenue that they will get from our partnership will be enough to convince them.
And do you know if they're going to have to change their marketing strategy at all to drive sales now that they're going to have this extra device as well? Did you talk to them about that at all?
Yeah, we haven't talked to them. We took just one of those pharmaceutical companies. We thought they were like the most fit for us. There's always a possibility that another pharmaceutical company is better or whatever. The thing is, they have a very extensive network of with medical professionals and hospitals. And I guess that's also how they promote their products. The thing is, we were planning on relying also on their network. We also have our network in with like key opinion leaders from UZ Leuven, the refractive epilepsy centers. But we don't think they will really change their sales approach because, it's a very specific market and they already have the network, I would say.
Okay, so they're perhaps trying to kind of retain their market share, but kind of save people money. So they might be able to charge more for their drug. Is that?
No, they will have like a lot more customers, using their drug because it's like a package deal that we have with the
So they still need to expand their customer base, or you think this will help them expand their customer base?
Yeah, this will help them expand their customer base. Yeah, they're more focused on other drugs, but they might want to expand on this drug as well.
Okay, go for it.
Yeah. Well, thank you for the pitch. I really like the structure and also, the structure and with the other pitch. And now it was also a little bit like the summary at the end. Really serves the purpose of a pitch. Thank you for that. I was wondering, what do you need at the moment to continue in the future? Because you had a clear timeline, well thought through.
So the most important thing is that we now laid our foundation for our biosensing, for our biosensor and our cartridge, but there still needs to be a lot of research done to optimize our product, to have a very high precision, and to make sure that it's as cheap as possible. What another important thing is that why we only launch our product later on, like you can see in our curve, it's like 3-4 years, is because getting this research done and getting it regulated takes a lot of time. We think it's around 2 or 3 years. And also a very important thing to mention as well is that for patients to use our product, and also for neurologists, it's important to them that it's getting paid back by the government or by insurance companies in the Netherlands, for example. In Belgium, it's the government that regulates it. But this also takes time. Like they need those clinical studies that prove they will definitely make a change. They need those market studies. They will have an economic impact. So you cannot just like push your product on the market like easily like that. It really takes a lot of time.
I also read that you did quite some customer validation actually, like stakeholder validation at least. What was if you look at the patients, was there something that that you did not know before you talked to them? What was your main takeaway?
So we had the luck to be able to talk with the CEO of Happy Hunter. Happy Hunter is like he started this company because his son has a very severe case of epilepsy, and he talked us through like the whole journey of him as a parent with the mother of how difficult it was because they spent so much time going to the hospital and back. They used so many treatments, and it really took a toll on the mental health of his kid. But what we realized is that as a child or like a person with epilepsy, you want to live a normal life. Like you don't want to have those attacks at any moment or anything. And it's possible that some attacks will never be solved. But for example, with his device, it's possible that people around him know that his child has a attack, like silent attack, that and so they know, and in that way, that's what they want to solve. Like you will never be able to. You need to cope with the disease. It will never go away eventually.
Okay. Thank you. I also hear like actually a lot of practicalities, which is a good thing, of course. Did you also dive in a bit to the like adherence, medication adherence?
What do you mean by medication adherence?
Well, that they take it, actually, the medication?
So valproate is the most used drug for epilepsy at the moment. I mean, we have if you see our, we made like a whole calculation. So in our appendix, you can see how many people are using valproate, for example, in Belgium. And like almost in all European countries, it's the same ratio, let's say. So based on that, we calculated how many people will probably use our product. And it's far enough that...
Did you also focus on that they are actually going to use it? Because you see a lot that this is practicalities, of course. You make it easier to measure for them. But there's also a big problem that the medication is there, but people are not taking the medication and not taking it at the right time. So how did you consider medical adherence, that they actually take it, and don't forget about their medication? Is that something you implemented as well?
At the moment, we didn't, but I would say this is like a very important relationship between the patient and the doctor. But what we could do, if eventually we will start with our device at home device, is that there is some kind of application that will tell them you should take your medication, for example. But yeah, the reason why we haven't thought about it is because we know that an at-home device is not feasible at this moment. Like, it will be in the future, but the people are not ready yet. For example, we talked with the neurologist of UZ Leuven, and he wasn't a fan of the idea because it happens a lot that they call the doctor for like very small things, and he loses a lot of time because of that. And we're afraid that when they have the device at home, they might see like a little change in concentration, or they might think, oh no, it's like a big problem to them because they don't know how to read it. And they might call the doctor for like just small things. But yeah, the future is that when they have the device at home, we will try to implement them to make sure that they have a notification to take their medication, for example. But yeah, we didn't think about it.
So it's on your list, but not on top of the priority right now.
Yeah, exactly.
Then I think that was it for this discussion. We have to move on to the next one to keep it in time. So I want to thank you. And then the next discussion is from Sense. So that will be played now.
Thank you very much.
Thanks.
Greetings to everyone. We are Sense, the RWTH Aachen University team in Germany, presenting the translational potential pitch. Stakeholder desirability. We have 4 points towards this. Studies have shown that excess valproate in the bloodstream causes severe poisoning associated with coma, depression, cerebral edema, and hemodynamic instability, and majorly teratogenetic side effects, making it particularly unsuitable for women. There are tests in the market to detect valproate, but we see that it takes a long time for the results to be seen. Here are the ELSA and HPLC-MS tests, not only requiring large volumes of sample, but are also quite labor-intensive. The other alternative, Cobra Integra FVALP tests, are highly costly. Also, we see that a lot of tests are taken unnecessarily. Reasons not specifically targeted for overdose, causing a resource waste. Moving on to business feasibility, splitting this into 2 parts. For technical development, we are currently in the prototype development stage. What mainly remains is the development of the user interface that enables the measured values to be displayed on a mobile app, for example, for easy reading. After which, we would form a contract with electronics companies to help us design and manufacture components and help us with the packaging. Entering the market. After a prototype is ready and we have proved it suitable for valproate detection, first would be to patent our model. Then we would certify the device, for which help from a specialized consultation firm would be taken. Towards sales. Hiring additional talents would be required, and we would establish contact to potential customers. Now towards the financial viability. Approximately 40,000 fertile women in Germany currently taking valproate are severely affected by side effects. To give you an estimate cost for a device, there would be a one-time investment cost of readout system costing approximately 600 euros. This consists of the potential stat module, 500 euros, microcomputer, display peripherals, 80 euros, PCB components, soldering, approximately 5 to 6 euros, packaging, 15 euros. The estimated cost of a single-use cartridge would be 53 cents, consisting of the IDE chip, 13 cents, MOF deposition for 10 cents, bonding and packaging, 30 cents. Finally, we show you the revenue model. Fixed costs incurred would be 40,000 to 60,000 euros, consisting of the additional development, 20,000 to 40,000 euros, patenting and counseling, 500 euros, medication device certification of Class B, 15,000 euros, and the set price would be for the readout system, 600 euros, and the cartridge for 2 euros, which we approximate to have a 1 euro profit. So the bare profits, assuming daily tests with 40,000 customers, would be 14.4 million euros. Well, so we say, epilepsy stopping you from living your dream life? Not anymore with your very own VPA measurement module.
Okay, thank you very much. Accents. I do have actually 2 questions. I don't know if someone else first wants to start.
I was thinking we could see if anybody from the audience has a question, Claire.
I'm looking at the live stream right now, and I don't think there are any questions. So.
And from the other teams?
If you have a question, please unmute yourself, and then it may be asked.
Okay, Claire, go for it.
Oh, okay.
We gave everyone a chance.
yeah, sorry. Well, this was the first time I saw your pitch as well. I saw another pitch before. I did read your TRD document, and there I also read. So you have a B2C product, right? B2C, right?
Sorry, I didn't understand the last part of the question.
Oh, you have a B2C product. You sell to the patients, right?
Yeah.
I read in your document, and I actually was a little bit looking for it in the pitch, that adherence, again, that there was no wide practice on the routine monitoring yet. And I wondered how you are going to change this behavior with your product.
Yeah. With our product, one of the goals would be to also deliver it to researchers so that they can actually get more data and more experience on how much use there actually is in monitoring VPA. As mentioned before by that sir, for example, we don't really know at the moment how the blood level is related to the actual VPA concentration in the brain. So researchers could use the tool to get more experience how this daily or frequent monitoring concept could actually improve medication.
Okay. And another question was, you were very detailed in the finances, which is good, but where do you have any cross references? How did you came up with the numbers?
Numbers, for example, the parts were basically based on how we bought them, for the price we bought them. For example, for production, we looked at, for example, online offers that are available for similar tools, which we took as an estimate. And then for things like packaging or the deposition of the MOF, which is the central part of our sensor, we basically estimated this ourselves. And yeah.
Okay, thank you.
Okay, hello, if I may maybe cut in. My name is Dariusz Wiśniewski, and I represent the Institute of Biotechnology and Molecular Medicine. And I have a quick question on, did you have any experience on talking with actual customers about the price that you estimated for the device alone and the cartridges as well?
So far, we don't actually talk about the price with customers, at least in my experience. We know that the actual sale price of the module itself is quite high, but we took the actual module or the actual, for example, machines used in hospital laboratories as a comparison, and in that regard, it's actually then again very simple, but it's a completely different use. So,
yeah. So, like one of the things we suppose why daily monitoring is probably not practice is just because of the high price of the tests. So, with the chemical analyzers that are currently being used, one test costs about €5, and including like factoring in all the other costs like running costs of maintenance and cost of operation of the devices, that's like around €10 per test. So we were thinking, on the one hand, if we can reduce the price significantly, then maybe that might change the attitude towards daily monitoring. So maybe the upsides are considered more than the financial downsides. But the other thing is that, of course, like these devices they already have. So our device will be quite a lot less expensive, but like these devices are already present and they have many different applications. So the laboratories have them anyway. That's why we're kind of thinking about focusing on the direct-to-consumer segment. So these people would actually benefit from having them at home, and like the portability and user friendliness and not having to go to the hospital provide the greatest utility for the customer in that regard.
Okay, thank you. And if it goes for the communication of the results of the test to the actual medical professionals, have you talked about the, it was beforehand, it was mentioned in another presentation, but have you actually talked to a neurologist on how he sees the communication and interaction if it goes for the acquired data from your device?
Go ahead. We talked with a local professor for epileptology, and she said that she's not really the fan of like this direct communication. So basically that patients read out the data and then like call the doctor and communicate. And with that, it would probably be better to just give an estimate and tell the customer then, or the user, okay, this is fine, or okay, you might be in a danger of an overdose, and then make the raw data, the raw concentration available, and then they can bring that data to the doctor and discuss the further approach with them.
Okay, thank you. I have one last question. If it goes for the for potential fundraising, do you have a plan for fundraising your, let's say, production for the whole system?
As it currently stands, I don't think we really have that. So like the thought was, if we want to commercialize this, the first thing we have is a working prototype, and then we can work from that. Like that is the most compelling argument, having like a functioning device. So our plan was basically to focus on that first, and then see if we can find investors for that. Maybe with a more elaborate plan then.
Okay, thank you.
Hi, Team iSense. I had a question about again about the customers. So obviously, most products that are sold to direct to consumers have a kind of a big marketing campaign behind them to raise awareness. This is kind of a brand new thing for people to buy. Two-part question. Have you thought about your marketing? And the second question is, how many customers do you think you're going to sell to in the first year, second year, third year? Because in your document it sounds like you're just going to get 100% of the market.
No. Like it was just to supposed to, like, this is the potential. This is what you can get if you get 100% of the customers, which of course is not going to happen. Just to show the potential. So I mean, like several aspects are quite bleak if you think about them, because in recent times prescription of valproic acid has been declining, which is not that enticing for a business to say, okay, this medication gets prescribed less and less, and this customer group, which we list as our core customers, which have the greatest benefit, actually it keeps like the advice from in Germany, at least, from different like government bodies and other institutions, are to prescribe this less to women in the fertile age, because it has these teratogenic effects which are very undesirable and should be avoided at all cost. So we were thinking that, like, for the people who still have to take valproic acid, where there's no other way than having this medication or a combination of different other medications, that for those we can still provide like this dose adjustment. So since there are some studies which kind of relate the severity or like the probability of these teratogenic effects with the aptness of the dosage, so if we can kind of regulate it down. But yeah, like this kind of I guess difficult to estimate how much of the how deep into the market we can actually penetrate. So I mean 40 million for the entire market, even at 10%, that's like still 1.4 million. So, and there's still the prospect of expanding outside of Germany. But yeah, I don't know. I think when it comes to marketing, I think we would really focus on this core customer group and kind of really stress the benefits. You know, I mean, if you can avoid the crippling effects of these birth defects to your future child, then would you not do this extra step and like get such a device? And then if it's like 2 euros a day or something, I think that's definitely worth it.
Okay, then I think it's time to cut off this discussion as well. I would really like to thank especially Stuart, Claire, and Darius for being in the jury. And then now in 5 minutes, the keynote lecture in the general will start, and afterwards the closing, 10 to 4, with the award ceremony. So I hope to see you there. And yeah, thank you to you all.
Thank you very much for
And
your
well
time.
done to the teams. Thanks.
Yeah.
Thank you.
Absolutely, it was great.
Nice, good to hear.
Bye.
Bye-bye.